Hello everyone. Today we are going to start with general embryology, and the topics that we are going to discuss today, they are the duration of the pregnancy, the general timeline, that is the pre-embryonic, embryonic and the fetal timeline, right? The primordial germ cells, very important. So in short, they are called as PGCs or the primordial germ cells, right? We will discuss clinical correlation with the primordial germ cells. These are the craniopharyngeal teratoma and sacrococcygeal teratoma. We will discuss about mitosis and meiosis, that I suppose you have already read it +2. Apart from that, the abnormalities which may occur during meiosis, and that is non-disjunction, and non-disjunction, it is the trisomy and the monosomy that we will discuss. So in trisomy, it is the Down syndrome that I will discuss, and in monosomy, it is the Turner syndrome. So now let's start with the topic, embryology. Now what is embryology? It is the study of formation. It is the study of formation, development and maturation of embryo. Okay, it is the study of formation, development and maturation of embryo. So this is called as embryology. Now the duration of pregnancy, remember the duration of pregnancy, it is about 9 calendar months. According to calendar, it is the 9 calendar months and 7 days, right? Or it can be 280 days, or it can be 40 weeks. Now the duration of pregnancy, it is calculated from the first day of the last menstrual period. Okay, it is calculated from, so it is calculated from the first day of the last menstrual period. So this is also called as the gestational age. Okay, this is also called as the gestational age. Now, the embryonic period, the conceptus is said to be an embryo in between the 3rd to the 8th week, right? So embryonic period, it is from 3rd week till 8th week, and the conceptus is said to be fetus between the 9th week till birth. So pre-embryonic 0 to 2 weeks, embryonic period, it is between 3rd to 8th week, and the fetal period, it is from 9th week till birth. Now primordial germ cells. Now these primordial germ cells, these are also called as the primitive sex cells. These are also called as the primitive sex cells, right? Now these primordial germ cells, they arise from epiblast, right? They arise from epiblast. Now what is this epiblast? Epiblast, it is also known as the primitive ectoderm. It is also known as the primitive ectoderm. Now primitive ectoderm means it will form the ectoderm later on, but right now it is not the ectoderm, it is something which will form the ectoderm, right? So in the 2nd week of embryogenesis, so remember that in the 2nd week of embryogenesis, when embryogenesis is taking place, this epiblast will give rise to primordial germ cells. Primordial germ cells, also called as the primitive sex cells. Now, these primordial germ cells, they are derived from epiblast. So, this is something that you have to mention. They are derived from epiblast. Now, remember that there is no genital ridge in the second week. We don't have any mesoderm before the third week. It is now the second week of embryogenesis. There is no genital ridge, there is no mesoderm. So everything will come in the third week. So it is the genital ridge or the mesoderm which will give rise to the testis and the ovary. So right now these primitive sex cells, since they don't have any genital ridge or no testis or ovary, what they will do? They will migrate. Okay, they will migrate to the wall of the yolk sac. They will migrate to wall of yolk sac, and it is by 4 weeks. By 4 weeks, they will migrate to the wall of the yolk sac by the 4th week, right? And again in the 5th week, these primordial germ cells, these PGCs will again migrate to genital ridge. They will again migrate to genital ridge. Genital ridge, it is also called as the gonadal ridge. It is also called as the gonadal ridge. Now this genital ridge or gonadal ridge, now this will differentiate and it will differentiate and it will give rise to the spermatogonium and oogonium. Okay, oogonia or oogonium, spermatogonia or spermatogonium, right? So yes, they will give rise to spermatogonium or spermatogonia or oogonium or oogonia. So the primitive sex cells are these primordial germ cells which will form the sex cells, that is the spermatogonia and the oogonia. Okay, so that is again in the 5th week when they will migrate to the genital ridge, right, the gonadal ridge. Till now, if you don't know what is yolk sac, what is gonadal ridge, don't worry about that. In the upcoming video, you will be like very clear, you will be crystal clear with all these terminologies. What is epiblast, yolk sac, these gonadal ridge, genital ridge, everything will be clear. As of now, just remember, there is something called as the primordial germ cells which will differentiate to form the sex cells, that is the spermatogonium and the oogonium. Now, two clinical correlations that are very, very important with respect to this, that is the craniopharyngeal teratoma and sacrococcygeal teratoma. Now, what is this? Now, you already know we have the primordial germ cells. Now, these primordial germ cells, or these PGCs, if they don't migrate to the genital ridge, now they have abnormal migration, or they reach the neck region, okay, and these primordial germ cells, they reach the neck region, and because of the pluripotency of these germ cells, okay, they will differentiate or they will lead to teratoma. It will lead to teratoma. It is basically a tumor of the germ cells. It is the tumor of the germ cells. So there is abnormal migration. Now these primordial germ cells, in some cases they can migrate the neck region. Now you already know craniopharyngeal. The name itself suggests that it is in the neck region, the pharynx and the cranium part, right? So in the neck region, basically there will be an outpouching which will develop as a result of these germ cells. Pluripotency means the ability of the germ cells to form all the three germ layers, right? Pluripotent, they have the ability to divide, okay, and that is why they can lead to teratoma or tumor of these germ cells. Apart from that, there could be one more abnormal migration. So if the primordial germ cells, or PGCs, if they reach the area or region between the sacrum and the coccyx. So you already know we have the sacral vertebra and the coccyx vertebra. So there is a region that is in between the sacrum and the coccyx, and in that region, if these primordial germ cells will migrate, they can lead to teratoma. So they reach the area between the sacrum and the coccyx, and because of the pluripotency of these germ cells, it can lead to teratoma. And it is termed as sacrococcygeal teratoma, which you can see over here in this particular picture how nicely it is given. See this outpouching, it is like outpouching, it is the primordial germ cells which have divided and like they have caused tumor over here. So this is called as sacrococcygeal teratoma. Now there are three questions that you have to answer. Now the very first question says the primordial germ cells migrating into genital ridge are coming from the wall of the yolk sac. I have already told you, yes, they are coming from the wall of the yolk sac. But again, if the question says the primordial germ cells are derived from, they are derived from epiblast, that is also called as the primitive ectoderm, okay, it is also called as the primitive ectoderm, and these primordial germ cells will give rise to spermatogonium and oogonium. Now, coming on to the difference between the mitosis and the meiosis. As you already know, mitosis is the one in which the chromosome number remains the same. The chromosome number remains the same. For example, this is a cell, right? This is a cell. It is containing 46 chromosomes, right? If mitosis takes place, the resulting cells will also contain the same number of chromosomes, that is 46 and 46, right? Now, in meiosis, we already know the meiosis is of two types, meiosis 1 and meiosis 2. Now, meiosis 1, it is also called as the reductional division. Why? Because the chromosome number is reduced to half. For example, if this cell is containing 46 chromosomes, then if the cell is undergoing meiosis 1, then the resulting cells, okay, the resulting daughter cells will have 23 chromosomes each, right? So the number of chromosomes is reduced to half. That is why meiosis 1, it is also called as the reductional division. Okay. Apart from that, if we talk about meiosis 2, so meiosis 2, it is equational division just like mitosis. Equational division just like mitosis. Now, if again there is mitosis, sorry, if again there is meiosis 2 in this particular cell, what will happen? The resulting cell will have 23 chromosomes each, right? The resulting cell will have 23 chromosomes each. Why? Because meiosis 2, it is the equational division just like mitosis. Apart from that, remember the stages of, yes, remember the stages, it is prophase, metaphase, anaphase and telophase. We have PMAT like prophase, metaphase, anaphase and telophase. So these are the 4 stages that I hope you already know from your +2 knowledge, right? Now coming on to the next part, that is the non-disjunction. That is one of the clinical aspect related with meiosis. Now in the non-disjunction, remember it is the failure of, let us write down the definition first. It is the failure of two members, right? It is the failure of two members of a homologous, or I can say a pair of homologous chromosome chromosome to separate during first meiotic division, and thus they pass together to one of the daughter cells, one of the two daughter cells that are formed, right? One of the two daughter cells. Now let us understand this, right? Now what are the results that you have to see over here? See the results. What will happen due to this non-disjunction? If these two members of a pair of homologous chromosomes, they don't separate, okay? Now if they don't separate, the two members will go to one cell directly and they will not go to another cell, right? So what will happen? One daughter cell will have extra chromosome, okay, extra chromosome, that is 24, right? And on the other hand, the other daughter cell will have one chromosome less, right? It will have one chromosome less. So you already know 23 minus 1, it will be 22. So one will have an extra chromosome and one will have one deficient chromosome, right? Now, if the fertilization occurs, now suppose if fertilization occurs with normal gamete, okay, if the fertilization occurs with normal gamete, right, with the normal gamete, I mean the gamete which is carrying 23 chromosomes. So it is a normal gamete, right? Now what will happen? This 23 chromosomes will combine with this daughter cell, right? This normal gamete will combine with this daughter cell and they will result in the formation of the zygote, which will contain 47 chromosomes. It will contain 47 chromosomes. Now you can easily see that one chromosome is extra. So that is something which is referred as trisomy. Okay, this is something which is referred as the trisomy. Now one chromosome is present in a triplet, right? Instead of being double, it is in triplet, and that is why you see the triplet, it is more commonly seen in the trisomy of chromosome 21. Okay, it is the trisomy of the chromosome 21. Yes, it is the trisomy of chromosome 21, and this is something which results in the Down syndrome. This is something which results in the Down syndrome. The infant will have 47 chromosomes, right? The Down syndrome, right? Apart from that, the other end, right? The other end, you already know the, now let us again the same situation. If the fertilization occurs with the normal gamete, if fertilization occurs with normal gamete, now normal gamete, it means 23 chromosomes. What will happen? This 23 chromosomes and this 22 chromosomes will combine with each other, right? So the resulting zygote will have only 45 chromosomes. Ideally it should be 46. Now it will have 45 chromosomes, one chromosome less, right? One chromosome less, and this is something which is called as monosomy, right? One chromosome is present in single form instead of duplet, right? So remember, it is the monosomy of the X chromosome that is more relevant, monosomy of X chromosome, right? Now let us suppose that the ovum, it is having no X chromosome, right? It is having no X chromosome and it is fertilized by sperm which is having X chromosome. Sperm में क्या होता है? XY होता है ना? So if it is fertilized by the sperm carrying X chromosome, then it will result in the formation of XO type, right? XO type, the zygote will be XO type. क्योंकि ovum में तो X था ही नहीं, there is no X chromosome, X is coming only from the father, so it will result in the formation of XO type, right? Or XO zygote, and this is something which is called as the Turner's syndrome. This is something which is called as the Turner's syndrome. So we have two diseases over here that we have to discuss. One is the Down syndrome and the other one, it is the Turner syndrome. Both of them are resulting from the non-disjunction of a pair of homologous chromosome, that is during the first meiotic division. That is again an important line that you have to remember. It is during the first meiotic division, right? So this is like the takeaway point from this slide. Now let us discuss these disorders one by one. Now, coming on to the Down syndrome, you already know that this Down syndrome is resulting from the non-disjunction, and this non-disjunction, it is occurring at the level of meiotic 1, right? So, yes, this is a case of non-disjunction, and this is taking place at the level of first meiotic division or meiosis 1, right, when the pair of homologous chromosomes don't separate, right. So that is, you have to remember. Apart from that, yes, it is associated with increased maternal age. It is associated with increased maternal age, right? If the age of the mother is more likely the chances that the baby is going to suffer from Down syndrome, right? So, yes. Now, remember that this is the most common chromosomal disorder in humans. And it is the most common cause of inheritable cause of mental retardation. Now, you already know mental retardation. It is also known as nowadays intellectual disability, right? So, it is the most common inheritable cause of mental retardation, right? So, the genetic cause you already know, it is the non-disjunction which is taking place during meiotic 1, right? Apart from that, you can remember the clinical features of this disease. With the help of mnemonic, child has problem. Now, what you will see over here, you will see cardiac defects. So, there are congenital cardiac defects, right. We can see hypotonia. Hypotonia means decrease in the muscle tone, right. You can write it also, it is decrease in the muscle tone. Apart from that, you can see there is an increased gap between the great toe and the second toe, right. So, there is an increased gap between the great toe and second toe, right. You can see over here leukemia, the cancers, right. You can also see duodenal atresia. In patients of Down syndrome, you will see duodenal atresia. It simply means that there is an obstruction in the duodenum which will not allow the food to move down, right, to move distally, atresia, blockage, right. Then you will see one disease that is called as Hirschsprung disease. Apart from that, you will also see Alzheimer's disease. And you will also encounter Simon's crease. there is only one crease in the hand, right? There is only one crease in the hand, or I can simply say there is single palmar crease. There is single palmar crease. Apart from that, if we talk about the problem, I mean the other clinical features that are related with this, most of these patients will have protruding tongue. Protruding tongue. The tongue will be coming out. Apart from that, you will see the rolling of the eyeball. Yes, the rolling of eyes. You will see that their occiput is flat. Occiput is flat. We have brush field spots. Brush field spots. Then we have low nasal bridge. We have epicanthal folds, like we can see epicanthal folds. And we can also see Mongolian slant. So these are the clinical features that you have to remember regarding this Down syndrome. So yes, apart from that, you have to remember that the chances of having second baby in Down syndrome are much lower, right? So yes, that is you have to remember. The two tests that are important, right? The two tests that are important, or I can say the screening test in pregnancy, if you want to encounter that whether the female is having the child, whether it is suffering from the Down syndrome or not, there are two tests in the screening where you screen for the disease. One, it is the triple test. And remember, in the triple test, what you will see that the alpha fetoprotein, it is decreased. The alpha fetoprotein, it is decreased. The HCG, human chorionic gonadotropin, okay, it is increased. In the mother, okay, in the mother, what you will see, the alpha fetoprotein, it is decreased. The human chorionic gonadotropin, it will be increased. And remember, the estradiol, it will be decreased. So the only content which will be, the only parameter which will be increased, it is HCG. Now there is another test which you can perform, and that is known as the quadruple test. Now this quadruple test, it is nothing but the triple test plus inhibin alpha. Inhibin alpha is increased more in amount, right? So we also see the levels of the inhibin A, inhibin alpha, right? So the levels are increased. So quadruple test is nothing but the alpha fetoprotein, HCG, estradiol plus the levels of inhibin, right? So yes. Now coming on to the next disorder, that is the Turner syndrome. In Turner syndrome, remember the karyotype. The karyotype, it is 45XO. The karyotype, it is 45XO. Yes, the gonads will be ovaries, and these are called as streak ovaries. Now when I use the term streak, it means the fibrous tissue is there. Fibrous tissue is there, it means that the ovary, it is hard, right? So yes, remember that these ovaries are not malignant. They are not malignant, and they are not to be removed during any surgery. They are not to be removed, right? They are not malignant, they are not to be removed. You already know the ovary is underdeveloped. See, for the proper development of the ovaries, both the X chromosomes are required, and over here only one X chromosome is there. So these ovaries are underdeveloped ovaries, also called as the streak ovaries, right? So you already know the estrogen will be less. The estrogen will be less, and this estrogen, right? This estrogen, if it is less, you already know the breast will be absent. The breast will be absent, right? And you can also see that there are 99% of the cases which are like aborted. So Turner syndrome, according to the latest data, it is 1 in 2500 pregnancies. 1 in 2500 pregnancies, right? Or 1 in 2000 live born females. So it's better if you write like this only. It is 1 in 2500 or 1 in 2000 live born females, right? So yeah. Apart from that, remember the other clinical features that are associated with it. You can see very well that we have the cardiac defects. Now, which cardiac defects? It is the bicuspid aortic valve, or it is also the coarctation of aorta. Okay, it is the bicuspid aortic valve, okay, or it is also coarctation of aorta, right? So cardiac defects are more common to these patients. Apart from that, you can see that their hands, okay, the hands and the foot, the feet, the hands. So there is something which you will see that is called as lymph edema. Lymph edema. Lymph edema, basically it is the edema which you can see over like the hands and feet will also be involved. Hands and feet will be involved, right? Apart from that, you can see the ovaries over here. The ovaries will be streak, okay, streak ovaries will be there, or I can say underdeveloped ovaries, or why we say is that streak ovaries, it is because of the fibrous tissue. Underdeveloped ovaries because of the fibrous tissue which is there, right? Apart from that, you can see the nipples, okay, the nipples over here, they are widely spaced, they are far apart from each other. Yes, these nipples, right? So nipples are widely spaced, right? The nipples are widely spaced, and the female, it is short stature, okay, it is short stature, female, short height, right, short stature, due to mutation in, that you have to remember, S-Hox gene. Due to mutation in the S-Hox gene. And apart from that, you can also see webbed neck. You can also see webbed neck, right? Webbed neck. It is the low neck hairline, right? It is the low neck hairline. The low neck hairline, which you can see over here. Now, you can also see, yes, the cubitus valgus. Yes, the cubitus valgus, right, cubitus valgus, and there is something which is called as the shield shaped chest. Shield shaped chest. Their chest is like shield shaped, okay, because of poor development of the ovaries, poor development of the breast, right, the absent of the breast, so it is like appearing like the shield shaped chest like appearance is there. So now what we can do in these patients, the management that you have to remember over here, the management, you have to give estrogen alone for 1 year to these patients, okay, and this estrogen you are giving for breast development. Now these female can conceive with the help of IVF, right? These female can conceive with the help of IVF, in vitro fertilization, right? And after giving her estrogen for 1 year, you can give, it is followed by estrogen and progesterone subsequently. And if it is like the pubertal age, then you can give her growth hormone, and it should be given like before 8 years of age. So this was about the Turner syndrome. So we have discussed about the duration of pregnancy, the general timeline, the primordial germ cells, the craniopharyngeal and sacrococcygeal teratoma, mitosis, non-disjunction, and Down syndrome and Turner syndrome. Thank you.